I'm going to go ahead and get started. This second session is really trying to look into some more translational aspects and some of these physiologic things that are associated with glaucoma. And the session is drug repurposing in glaucoma. And the first person to speak will be Lou Pasquale, who you all know. And so we'll need no further introduction. I will make one comment. I'm dressed really relaxed because, you know, think tanks, I just think better when I'm in like my regular clothing. But I will wear a suit tonight to honor Lou.
Thank you, Richard. So the drug repurposing session is going to be focused more on glucose metabolism and insulin signaling. So I'm going to lay the foundation for this. I tried to get Adriana DiPolo to give this talk, but she had other things going on. So I'm taking it over. And here are my financial disclosures, and the outline we're going to go through is review insulin production and metabolism, review insulin signaling in tissues relevant to glaucoma, and then talk about insulin resistance, what it is, and how it is related to glaucoma. I will leave the tackling of the therapeutic part of this to my co-speakers in this session. So insulin production and secretion, as we all know, is in the pancreatic beta cells. The insulin gene is small. It's only 3 exons. And some believe that the small size of the insulin gene puts the humans at a disadvantage because it cannot adapt to the modern world where we have abundant available foodstuffs, and that somehow makes us all, at risk for getting type 2 diabetes. So this slide just reviews insulin production and secretion. We start out with pre-pro-insulin with the alpha and beta chain connected by a C peptide, and in the endoplasmic reticulum, we get removal of the C peptide, and you see down below the insulin molecule, which actually I want to point out also has a very short half-life. And so that is the sort of overview. When you start talking about insulin signaling, it's really quite complex. It has to do with many things that transcend glucose uptake, and including glycogen synthesis, gluconeogenesis, liponeogenesis, and we heard in the last session how lipids are important in glaucoma. We just only touch the surface of that subject. Protein synthesis, gene expression, DNA synthesis, amino acid uptake, apoptosis, and autophagy. And some of the processes involved span from glucose homeostasis to cognition and lifespan. So many of the biochemical events triggered by insulin binding to cell surface receptors might be good for glaucoma. And so we're going to cover this in a little more detail. So insulin is diffusely found throughout the human retina. This is a study from 1984 demonstrating with an anti-insulin antibody that basically you can find insulin throughout the whole retina. Insulin receptors are also located throughout the entire retina. This is a paper from 2009. Insulin receptors, I just showed you, are basically diffusely located throughout the posterior segment of the eye, but they're also located in the anterior segment of the eye as well, in the cornea, in the ciliary body epithelium, in the lens capsule. So insulin receptors are located throughout the eye, so they must be playing an important role in ocular processes in general and in glaucoma in particular. So I want to now focus about insulin resistance. What does this mean? And on the macro level, it's pretty simple. You get into this sort of vicious cycle where you feel tired and hungry, you eat a lot of food, you make a lot of insulin, but then you make so much insulin that the cells become basically immune to them, and the insulin signaling stops, and sugar gets stored in fat, and then the cycle repeats itself. And it's thought that 1 in 3 adults have some degree of insulin resistance. But what does it mean on a cellular level? And this is where it's important in ophthalmology and in glaucoma. And you can see that insulin resistance might mean that, insulin will interact with the receptor, but nothing else happens inside the cell. And why might that be bad? Well, insulin signaling has to do with uptake of glucose into the cell. So if the signaling is abnormal, then your intracellular concentration of glucose will be low. This has implications for mitochondrial function because the mitochondria require glucose to run. They're like the little batteries inside your cells. Also, it allows a more pro-inflammatory milieu to develop, so it can contribute to neuroinflammation. So some very nice work by Adriana DiPolo showed that insulin resistance in retinal ganglion cells causes dendritic retraction, mitochondrial dysfunction, and apoptosis. And this is some of the data from her work in molecular neurodegeneration, showing that you get retraction of the dendritic tree that develops 2 weeks after experimental ocular hypertension in mice. So you raise the eye pressure in mice, and then you start to see dendritic retraction, and this is exacerbated by impaired insulin signaling. Insulin actually promotes dendritic regeneration in adult retinal ganglion cells after axon injury, as indicated by this data that, again, Adriana was responsible, a senior author on, and published in Brain in 2018. So also there are effects of insulin signaling on the supporting cells for retinal ganglion cells. So microglia are important for helping to maintain retinal ganglion cells, and microglial activation can propagate retinal ganglion cell loss in experimental glaucoma. And you could imagine, again, that if there's impaired insulin signaling, this allows the pro-inflammatory mediators to take over and contribute to apoptosis in the retinal ganglion cell. And so this slide kind of summarizes insulin resistance in microglia, because if insulin doesn't bind to the insulin receptor, or you don't get the inhibitors of pro-inflammatory mediators, you activate the microglia, and that will contribute to retinal ganglion cell apoptosis. And we heard in the last session how immune factors are showing up in GWAS in relationship to glaucoma. So what about the clinical evidence for insulin resistance in glaucoma? And in primary open-angle glaucoma, there is impaired retinal vascular dysregulation. And this is work that Colm O'Brien did 25 years ago, looking at forearm blood flow in untreated normal tension glaucoma. So if you give intravenous acetylcholine to a untreated normal tension glaucoma patient, there is failure of dilation of the forearm blood vessel, whereas you could see in the white circles in the control, this is normal. I would argue that this has something to do with insulin resistance. And so it is true that if you look inside the eye, there is a more complicated neurovascular unit where astrocytes get coupled to endothelial cells, and they supply glycogen to the endothelial cells. And if those glycogens are not transferred to the endothelial cells, you get reduced nitric oxide synthase activity, and nitric oxide synthase activity is important for inducing vasodilation, and ultimately this causes apoptosis in endothelial cells. Mitochondria also integrate glucose and lipid metabolism for energy generation. Insulin preserves the NADH-NAD ratio to maintain mitochondrial function, and we know from the work from Jeff Liebman and Simon John at Columbia that this is an exciting area where there could be interventional strategies to help our glaucoma patients. So it sounds like it's a perfect storm, right? Like impaired insulin signaling can lead to retinal ganglion cell dendritic retraction and apoptosis. It can activate microglial cells and lead to neuroinflammation. It alters neurovascular coupling and causes mitochondrial dysfunction. And all these things gang up on the retinal ganglion cell to produce glaucomatous optic neuropathy. So the evidence that insulin resistance contributes to open-angle glaucoma is quite strong. Interestingly, this is a study that was published recently in May of 2023, showing that intraventricular injection of a compound called S961, which blocks insulin receptors, produces a glaucoma phenotype. When you inject this intraventricularly, you get elevated intraocular pressure and retinal ganglion cell loss. That's on the basic science level. What about the clinical level? Here's studies that have been summarized up to the year 2014. These are case-controlled studies and meta-analysis indicating that type 2 diabetes is a risk factor for open-angle glaucoma. Here are some cohort studies linking type 2 diabetes to open-angle glaucoma, including the study that Jahi and I co-authored in 2006. You know, and one of the criticisms of these kinds of epidemiologic studies is that they're subject to detection bias. So you're more likely to be examining diabetics more carefully for glaucoma than non-diabetics, and that could contribute to these findings. And you can't argue with that, but every study shows the same thing, including studies that post-date this meta-analysis in 2014. But then you can invoke the genetic argument, and this is basically relatively free of detection bias. And so you can do something called Mendelian randomization experiments, where you look at the genes associated for insulin resistance and ask, are they associated with glaucoma phenotypes? And the answer is that they are. And so the genes for type 2 diabetes in these Mendelian randomization experiments are associated with primary open-angle glaucoma. And again, Anthony's work, this is really amazing. So hemoglobin A1C is a biomarker for insulin resistance, right? So the higher your hemoglobin A1C, that is an indicator that you're more prone to insulin resistance. And again, using the UK Biobank, he's demonstrating that even patients with basically a borderline high hemoglobin A1C. You know, when you go to your internist and he says, your hemoglobin A1C is 6.1, you just go, you know, my blood sugar is okay. But, you know, that was associated with a thinner retinal nerve fiber layer thickness in the UK Biobank. And again, that was associated, diagnosed diabetes was associated with a thinner retinal nerve fiber layer. And so this is among people who don't have glaucoma and don't have diabetic retinopathy. So therefore, this addresses the detection bias and tells us that insulin resistance is something we really need to pay attention to in the pathogenesis of primary open-angle glaucoma. So what are some of the signs of insulin resistance in pre-diabetics? This is hemoglobin A1C between 5.7 and 6.3, BMI in the overweight range, blood pressure greater than 130 over 80, fasting blood sugar greater than 100 milligrams per deciliter, triglycerides greater than 150 milligrams per deciliter, hemoglobin A1C less than 40 milligrams per deciliter in men, and less than 50 milligrams per deciliter in women. So I would argue that if you look through the literature, there's some evidence to suggest that some or all of these things are related to primary open-angle glaucoma. And so, again, adding more evidence that insulin resistance is a really important thing to be thinking about when we think about the pathogenesis of primary open-angle glaucoma. So in conclusion, disruption of insulin signaling causes an increase in IOP. Insulin signaling plays an integral role in retinal ganglion cell homeostasis. Insulin resistance is associated with glaucoma-related traits. And what I haven't talked about is that insulin resistance is also implicated in other neurodegenerative diseases, Alzheimer's disease and Parkinson's disease, and very high percentage of Alzheimer patients have type 2 diabetes. And enhancing insulin sensitivity may be a therapeutic target for glaucoma, and this is where my co-speakers will have some commentary. And I just want to acknowledge Adriana and David Calkins, who really have put together a lot of the thought processes here and have done a lot of the work. I think a lot more needs to be done in terms of on a basic science level regarding insulin signaling and its relationship to retinal ganglion cell health. Also want to acknowledge Jae-hee, who did one of the important studies regarding type 2 diabetes in relationship to primary open-angle glaucoma, and Anthony with that beautiful study on hemoglobin A1C and its relationship to retinal neurofibrillary thickness. Thank you for your attention.
Thank you, Lou, for that great overview. So this is going to lead into a couple of talks on some of the translational aspects relative to glaucoma. We're going to start off here now with Joshua Stein. Josh is the Dreyer Career Development Professor at the Kellogg Eye Center in University of Michigan. And we have Josh's slides up. And so Josh is going to talk about metformin and glaucoma.
Good morning, everyone, and thank you very much for including me in this program. This is my first think tank, and it's such an incredible group of scientists who've come together, and kudos, Lou, to you and the organizing committee for putting together such a great symposium. I'm going to be talking about a topic that dovetails Lou's talk, and this is some work we've done looking at associations between metformin and glaucoma. These are my financial disclosures. So caloric restriction and drugs that mimic caloric restriction are capable of lengthening the lifespan and reducing the risk of some age-associated diseases. They've been studied in diabetes, cancer, cardiovascular disease, and the neuroprotective effects of caloric restriction and caloric restriction mimetic drugs are thought to occur as a result of back-shifting gene expression profiles of an older individual, so they resemble that of a younger person who is either not on a restricted diet or are not taking these medications. And there are two known caloric restricting medications that are available in the United States. One is rapamycin, which is a macrolide antibiotic, and the other is metformin. Metformin is a first-line treatment for patients with type 2 diabetes. It's very effective at lowering blood sugar. It's pretty well tolerated with few side effects. It's widely available and relatively inexpensive. So if we're thinking about medications that could potentially be repurposed, it kind of checks all the boxes in terms of one that might be readily available and tolerable for this purpose. If you look at sort of the history of metformin, It was discovered back in the early 1900s, and it first got approved for treatment for diabetes in France in the 1950s, and then it took about 50 years before it got approved for use in the United States. Since then, we've seen not only a widespread use of metformin, but studies have shown that it's able to reduce the risk of cancer, reduce the risk of diabetes, and in some animal models, lengthen the lifespan. There are a number of different ways that or proposed mechanisms as to how metformin may be beneficial. The primary hallmarks include loss of proteostasis, telomere attrition, genomic instability, epigenetic alterations. There's dysregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, altered intracellular communication, and stem cell exhaustion. Based on some of this prior work and the fact that we're seeing protective effects for other chronic conditions that tend to affect older individuals, my colleagues and I were interested to see, could metformin potentially be neuroprotective in patients in terms of risk of glaucoma among patients with diabetes. So the questions we were interested in are drugs like metformin associated with a reduced risk of late onset diseases such as open-angle glaucoma? If so, is there a dose-response relationship? If you consume more of metformin, do you have more of a protective effect? Do you need to consume a certain amount of metformin to attain any beneficial effect? And then to try to tease apart, is the mechanism reducing or better glycemic control, or is the mechanism one of these other effects? We also wanted to look at other classes of diabetes meds to see if they would have a similar effect. So for this study, we used a large administrative claims database. These are patients with commercial health insurance in a large US managed care network throughout the United States. The database contained data, longitudinal data for 10 years on this population. The inclusion criteria were patients with diabetes, and we limited it to patients older the age of 40, and if they had pre-existing glaucoma, we excluded them. Here you can see sort of how we set up our analysis. We used the first 2 years the patient was in the plan as a look-back period. During that period, we confirmed that they had diabetes, that they didn't have glaucoma, and we quantified how much metformin they used. Then we followed them after the look-back period, and we monitored their ocular status, their hemoglobin A1C, how much metformin they were consuming, use of other diabetes medications, as well as other ocular and non-ocular conditions. And then we censored patients when either they developed glaucoma or their last visit to an eye doctor, which would presumably be their last opportunity to get glaucoma. We created 3 Cox proportional hazard regression models. The first model, we looked at metformin and quantified it in 4 quartiles, so 1 to 350 grams, 316 to 660 grams, 661 to 1,110 grams, and above 110 grams. And then we compared each of those quartiles to those who were not taking any metformin. The second model, we just treated metformin as a binary variable, less than 1,110 grams versus more than that. And then the third model, we treated as a continuous variable where we looked at daily use. We adjusted for a number of potential covariates. These included demographic factors, age, sex, race, geographic region of residence, socioeconomic factors like income and education, various other ocular comorbidities, systemic comorbidities, including hypertension, hyperlipidemia, a measure of overall health, the Charlson Comorbidity Index, and we also considered the type of diabetes. As a time-dependent covariates in our model, we considered ocular surgeries, use of other classes of diabetes medications, and hemoglobin A1C. So we had over 150,000 patients in this data set with diabetes and no glaucoma at baseline, and about 6,000 patients developed incident glaucoma during the follow-up period. You can see the length of time from the first eye exam to the last eye exam for those who did not develop glaucoma was about 4 or 5 years, and similarly, from first eye exam to first glaucoma diagnosis was about 5 years. Here you can see the medications that the patients were consuming. You can see metformin was the most commonly used anti-diabetic medication for this cohort. And you can see the percentage of patients who developed and did not develop glaucoma for each of the different medication classes. It's slightly less for metformin. This is a busy slide, but it basically shows the results of our primary model. Not surprisingly, we found that elevated age, higher age is associated with the increased hazard of developing incident glaucoma. We found that race was important. African Americans, Latinos, and Asian Americans had a higher odds or higher hazard of glaucoma. Various education and socioeconomic factors were not statistically significant. If you look sort of towards the bottom, you can see type of diabetes. Type 1 diabetes is associated with the reduced, 16% reduced hazard of glaucoma. Hypertension was associated with a 37% increased hazard of glaucoma, and this is work we and other groups have published in the past. And overall health was associated with a slight reduction in risk of glaucoma. After adjusting for all those other factors, these were kind of the key ones of interest. And here you can see for every 1 point increase in hemoglobin A1C, the hazard of glaucoma went up 8%. And then after we adjust for hemoglobin A1C, you can see the patients taking the highest quartile of metformin, which is 1,110 grams or more, had a 25% reduced hazard of developing open-angle glaucoma. The lower doses of metformin use were not statistically significant. Then we looked at the other classes of anti-diabetic medications, and you can see none of those medications were statistically significant. When we looked at the second model, we found those who consumed greater than 1,110 grams of metformin over 2 years had a 22% decreased hazard of glaucoma compared to those consuming less than that or no metformin. When we looked at daily use on a continuous level, every 1 gram increase in metformin was associated with a 0.16% reduction in glaucoma. The standard dose of metformin is 2 grams a day. So someone taking 2 grams a day for 2 years would have about a 21% reduced risk of glaucoma. Then we developed this fourth model, and this is a little, I'll try to walk you through it, how we did this one. The first step, we built a logistic regression model to determine a patient's underlying inherent risk of glaucoma, just based on their demographics, their socioeconomic factors, ocular and systemic comorbidities. And we rated everyone as in one of 4 quartiles. Then we looked at the highest risk and the lowest risk quartile. We then did a Cox regression model to determine the survival functions for the low and high risk individuals. And then we compared the risk reduction for patients stratified by hemoglobin A1C and by metformin consumption. So here you can see, again, the slide is a little busy here, but you can see there we stratified hemoglobin levels of 7, 10, and 13, and then low risk, low and high risk, and no metformin, moderate amount of metformin, highest amount of metformin. And you can see that the group that tended to have the largest risk reduction were the patients with diabetes who had the highest underlying risk of glaucoma, the worst glycemic control, and consumed the most metformin. So the key findings of our study: use of metformin was associated with a modest reduction in the risk of glaucoma among patients with diabetes. Those who took metformin at the standard dose of 2 grams a day for 2 years had a 21% reduced risk of glaucoma. There does appear to be a dose response effect where the patients consuming the most metformin had more of a risk reduction of glaucoma compared to the non-users. Every 1 unit of hemoglobin A1C increased the hazard of glaucoma 8%. And since metformin lowers hemoglobin A1C, improved glycemic control was probably helping to some extent, but because we didn't see the other classes of diabetes medications associated with reduction in risk of glaucoma, that led us to conclude that there may be something besides just glycemic control that may be responsible. This is another study. This is a smaller study in Bosnia that also looked at this, and they also found that metformin use decreased the odds of glaucoma as well as diabetic retinopathy. There's one study that did not show an association. This is a study in India, but you can see that they only had 5 or 8 participants that were on metformin and 28 that were not. So it was a pretty small study, and I don't know if it was sufficiently powered to answer the question. We're fortunate that Dr. Colm O'Brien is in the audience here. He and his lab have really done a tremendous amount of work in this area. They've published this great review paper on the antifibrotic processes and bioenergetics of metformin and glaucoma, and he's going to comment a little bit on some of the work he's been doing. This is a table from his review, and one of the potential mechanisms of how metformin may be beneficial is targeting the pro-fibrotic environment. AMPK activation is known to inhibit the pro-fibrotic TGF beta, and you can see that this role of TGF beta can impact intraocular pressure. It may impact the trabecular meshwork, the lamina cribosa, aqueous outflow. There's various different mechanisms both that as to how and why metformin may be beneficial. Another known effect of or another known mechanism is reducing mitochondrial dysfunction of cells of the lamina cribosa, optic nerve head, and trabecular meshwork. And as Lou mentioned in his talk, this may be important in glaucoma as well. How about beyond glaucoma? If we look at other chronic eye diseases, there's 2 studies showing that metformin use is associated with a reduction in risk of diabetic retinopathy. And there are 3 large studies showing that it may be beneficial in macular degeneration. Obviously, all these are different diseases of the eye, but they're all chronic and they're all associated with age. In terms of our study, the strengths, it was a large sample with a diverse population. The main limitations, it's a retrospective study, and because this is a large healthcare claims database, all the diagnoses were made exclusively using billing codes. There was no information on intraocular pressure, cup to disc ratio, findings from structural and functional testing, et cetera. And I'll talk a little bit more about that shortly. The study was conducted on patients with health insurance. Additional work would need to be done studying other populations. And all the patients in our analysis had diabetes. We did not try to explore the risk of glaucoma in patients who were not diabetic in taking metformin. And these days, metformin is being more commonly used in patients without diabetes. So there's more work that needs to be done in that area. In terms of future work, I'd love to see this study that we did replicated using a more robust data set that's got granular phenotypic and biomarker data, such as this source repository that Sally mentioned earlier. This would help us be able to tell, is any reduction in risk of glaucoma because metformin's affecting intraocular pressure versus nerve fiber layer versus changes on the visual field, to study associations between metformin and glaucoma among persons without diabetes, to leverage precision medicine to better understand how genetic environmental factors may modulate the neuroprotective effects of metformin on glaucoma, and then ideally to do a prospective randomized controlled trial that would definitively be able to answer the question whether a drug like metformin reduces glaucoma risk. If I was doing the study again, I would replicate it using SOURCE. This is a big data consortium that I've been spearheading. It's a bunch of academic ophthalmology departments, all of whom are on EPIC, and we're pooling our data for patients with eye diseases seen at our institutions. We're collecting demographics, all the structured data elements, the billing data, ICD and CPT codes, some patient-reported outcome data, laboratory test results like hemoglobin A1Cs, data from operative reports, systemic medications. And the data set has both granular ocular as well as non-ocular data. We've developed code to pull all this data from our EHR, and we're now sharing it with other sites who are also on EPIC so they can pull their data. We're working with a company to de-identify all the data so it's completely free of PHI. Then we're pooling the data, aggregating the data. We're linking in data from diagnostic tests. Hopefully in the future, we'll link in genomic data, and then making this pooled data set accessible to researchers in the consortium. We have 15 active sites that are sending their data in and are able to access and do work with SOURCE. Overall, there are about 4 sites that are in the process of either joining or have joined the consortium. So it should be a very useful resource to help us answer some of these questions in the years to come. We have about 4 million patients and 20 million office visits for eye problems and many lab test results to be able to answer these questions. And we're able to parse out very, very detailed clinical information like the findings that we document. I think, Richard, you had mentioned notching. You can go parse that kind of information out of the notes, so you can consider that in these analyses. And, for looking at cornea, we can parse out all these different types of pathology, so you can really nail down very detailed phenotype information. We're able to link in data from diagnostic tests. Where the other cool thing about sources, we're able to link in other pots of data, such as social determinant of health data, which I think is becoming more and more important for these analyses, link in mortality data, as well as community and neighborhood level data. So in conclusion, there is mounting evidence that metformin may be beneficial for reducing the risk of glaucoma. The beneficial effects of metformin may act at the level of the optic nerve, the nerve fiber layer, and/or the trabecular meshwork. The beneficial effects there are likely helping not only via improved glycemic control, but probably also through other mechanisms involving neurogenesis, inflammatory systems, or longevity pathways targeted by these caloric mimetic restricting medications. Metformin is a safe, effective, well-tolerated, and inexpensive medication, so it would be a great option to get repurposed if indeed future studies show it reduces the risk of glaucoma. Additional studies are needed to better understand how metformin is affecting intraocular pressure, as well as structural and functional changes to the optic nerve. And assuming future studies align with the existing literature, that may help us make a convincing argument for a large prospective clinical trial to really definitively answer this question. I'd like to acknowledge my co-authors on this study and the folks in my lab. Thank you.
Thank you, Josh, for that really thoughtful presentation. We're going to follow this up with Chi Tui. I hope I've said it right this time. She's at the University of Pennsylvania at the Shea Eye Institute and the Kirby Center for Molecular Ophthalmology. And she's going to talk about glucagon-like peptide receptor agonists as a possible novel therapeutic agent for glaucoma.
Thank you for that introduction. Good morning, everyone. I want to thank the conference organizers and in particular Lou for inviting me to talk today. I've already learned a lot from the previous session, which was excellent. So here's hoping we can give good follow-up. So today I'm going to be presenting our data suggesting that GLP-1 receptor agonists are a class of medication used to treat type 2 diabetes and may affect microglia and astrocyte reactivity and may be a viable target for glaucoma neuroprotection. Here's a list of my funding sources, and one thing I have not had a chance to update is that I just received a notice of award for R01, which is going to be supporting, thank you. I am very excited in case you can't tell, which is going to be supporting a lot of the work that I'll be presenting here. In particular, I do receive research funding from Neurally Inc., but that funding is not responsible for the bulk of the data I'm presenting here. Okay, so to start with, I'll talk a little bit about what GLP-1 receptor agonists are and what it is that they do. So GLP-1 is an incretin hormone that is secreted by the GI tract, and it regulates blood glucose, weight, and satiety, and does this by exclusively binding to the receptor. GLP-1 receptor agonists increase GLP-1 receptor signaling and is used initially to treat type 2 diabetes mellitus. And one thing I found interesting about this class of medication is that the prototype GLP-1 receptor agonist is discovered in the saliva of the Gila monster, which is pictured here. It's a really large and sedentary lizard that lives in the Sonoran Desert. So I'm from Tucson, Arizona, so there's sort of a nice link there, which is also a part of the Sonoran Desert. So the reason they secrete this particular agonist from their saliva is they can often go months and sometimes a whole year without eating, just because there's not that much food in the desert. So when they do eat, they need to secrete something to sort of jumpstart their pancreas, and that's where this comes into play. GLP-1 receptor agonists were first approved by the FDA in 2005, and it has rapidly become a very popular second-line agent for treating type 2 diabetes for a variety of reasons. First of all, both sub-Q and oral formulations are available, and dosing is relatively infrequent compared to other diabetic medications. It's typically once a day and sometimes once a week, and also offers cardiovascular protection and decreases all-cause mortality, specifically related to heart disease. Recently, it was also approved for weight loss in 2021, and I'm sure a lot of you have heard of these medications like Ozempic, so it all sort of falls in the same category of medications. So let's talk a little bit about why we care about GLP-1 receptor agonists, specifically in the setting of neuroinflammation and glaucoma. And this is all work done by Jacob Sterling, pictured here, who was an MD PhD student in my lab. So as we know, astrocytes are the most abundant type of glial cells in the CNS, and they play many important supportive roles. They support neural metabolism, and they support cellular function, and they also form the blood-brain and blood-retina barriers. And astrocytes, in a process called reactive astrocytosis, changes form in response to injury and disease. And this form change is sort of along a continuum. But specifically, a class of reactive astrocytes called C3 positive astrocytes, otherwise known as A1 astrocytes, have been found in Alzheimer's, Huntington's, and most pertinent to our talk here, Parkinson's disease. This is in contrast to the C3 negative astrocytes, which plays a more neurotrophic role. So these C3 positive astrocytes have been found to be neurotoxic in retinal ganglion cells after optic nerve crush in a mouse model. So basically, after optic nerve crush, in a study by Lidlow and Barris that was published in 2017 in Nature, they found that microglia releases a trio of cytokines, IL-1 alpha, TNF alpha, and C1q. And these cytokines will be important later. I'll come back to them. And these cytokines result in reactive astrocytosis and the formation of C3 positive astrocytes. And these astrocytes are responsible for retinal ganglion cell death. And they know this because by giving neutralizing antibodies to these trio of 3 cytokines, they were able to rescue retinal ganglion cells after optic nerve crush. So NLY-01 is an experimental GLP-1 receptor agonist that has good CNS penetration and a long half-life. It penetrates the blood-brain barrier quite easily to act centrally. And in a study that was published in 2018 in Nature Medicine, in a mouse model of Parkinson's, NLY-01 was found to prevent C3 positive astrocyte transformation. And they reduced the microglia release of IL-1 alpha, TNF-alpha, and C1q, so these 3 cytokines. And this resulted in neuroprotection, decreased the loss of neurons in the central nervous system, and improved motor functions in these mouse models of Parkinson's. So this is all sort of background information before we sort of pursued our own study. So this we know. What we also know is that GLP-1 receptor activation broadly induces downstream effects that are anti-apoptotic, pro-survival, and also are anti-inflammatory. So in this very nice review by one of the groups who first pursued GLP-1 receptor agonists for treating Parkinson's disease, what they found was that GLP-1 receptor activation inhibits NF-kappa B, and also inhibits activation microglia and pro-inflammatory cytokines such as TNF-alpha. And if you guys recall from Lou's talk earlier, this is the same pathways that was also inhibited by insulin binding to its receptor in the retinal ganglion cells. So now that we know NLY01 and possibly other GLP-1 receptors inhibits reactive astrocytosis, and that reactive astrocytosis results in retinal ganglion cell death after optic nerve crush, our question was, well, does increased intraocular pressure also result in a similar reactive astrocytosis? And could this class of medication, GLP-1 receptor agonists, and specifically NL1, result in neuroprotective effects? So we sought to answer this question by using the microbead injection model of glaucoma. So this is a model where you inject a microbead in one eye of a mouse, and the microbead themselves sort of make their way over to the iridocorneal angle and results in elevated intraocular pressure. And the other eye we inject with just basically balanced cell solution, and that serves as a control. So in our hands, once we do this injection, intraocular pressure began to elevate after about 5 days and remains elevated with 2 injections for about 7 to 8 weeks. So we injected both wild-type animals and also animals with triple knockout of these 3 cytokines that we know are important for reactive astrocytosis. After injection at various time points, we use magnetically activated cell sorting to sort out different populations of astrocyte Miller cells, microglia, and macrophages. And what we found was that in retinal cellular population enriched for astrocytes and Mueller cells, after 3 days, there was not an elevation in the genes associated with C3 positive astrocytes, C3 negative astrocytes, or pan-reactive astrocytes. But after about 14 days and then 42 days, we see that these genes associated with C3 positive astrocyte and pain reactivity was elevated in the wild type, but not in the triple knockout animals. Further, we saw elevated C3 mRNA expression and protein in the wild type, but not the triple knockout animals. So this tells us that these 3 cytokines are necessary and sufficient for the C3 positive astrocyte reactivity that we observed. When we took flat mounts of retinas isolated from animals injected with microbeads after 42 days, and we labeled for BRN3A, which is retinal ganglion cell marker, we saw that percent retinal ganglion cell survival in the bead injected eyes relative to the BSS injected eyes was relatively preserved in the triple knockout animals compared to wild type. When we looked at expression time course after injections, what we saw was that after about 7 days, the expression of IL-1 alpha, TNF alpha, and C1q began to elevate. What is interesting is that the elevation did not occur at the same time course in a macrophage-enriched population of cells versus a microglia-enriched population of cells. Specifically, the macrophage-enriched population of cells started to express these cytokines a bit earlier than the microglia-enriched retinal cellular populations, which did not begin to have elevated expression of these cytokines until about 2 weeks after injection. When we gave wild-type animals NLY01, dose 5 milligram per kilogram, injected sub-Q twice weekly, what we saw was that about 14 days post-injection, the cytokine expression began to elevate in a macrophage-enriched population of retinal cells, and about 42 days post-injection, they began to elevate in both a macrophage and microglia-enriched population of cells, but this elevation was decreased in the NLY01 treated animals. And this sort of dovetails nicely with the expression time course I just shown in the previous slide. Further, NLY01 reduced astrocyte reactivity, both at 14 days and 42 days post-injection. Similar for C3 mRNA expression as well. And finally, here is sort of the key slide here. NLY01 was shown to rescue retinal ganglion cell somas, a stain for the RGC markers BRN3A and RBPMS. What we saw was that percent retinal ganglion cell survival in the bead injected relative to the BSS injected eyes was essentially 100% preserved in NLY01 treated animals with elevated intraocular pressure compared to the eyes that had intraocular pressure elevation but was not treated with NLY01. Okay, so after this study, our question was, well, does this effect that we observe in NLY01, which is an experimental, not commercially available GLP-1 receptor agonist, also apply to GLP-1 receptor agonists that are FDA approved for treating diabetes and weight loss? So we decided to compare application of liraglutide with NLY01, and both were given sub-Q twice weekly. And we found was that, yes, so both liraglutide and NLY01 resulted in rescue of retinal ganglion cell somas and axons after about 42 days after microbead injections. We also wanted to know whether topical GLP-1 receptor agonists are a viable treatment option. As the sort of a first pass, we simply took liraglutide, which is a long-acting FDA-approved GLP-1 receptor agonist, and lexisenatide, which is a shorter-acting GLP-1 receptor agonist, and dissolved them in PBS. And then we simply gave these drops twice a day to animals after giving them microbead injections. We saw similarly, they did result in some degree of rescue of retinal ganglion cell somas and axons. Noticeably, however, the degree of rescue here was a bit less than with systemic administration. You can see it's about 25% for retinal ganglion cell somas, about 50% for the axons, and this is compared to the near total rescue that I showed in the previous slide. Okay, so now we know that GLP-1 receptor agonist results in functional preservation of retinal ganglion cell somas and axons. What about retinal ganglion cell function? Well, to sort of answer that question, we decided to use multi-electrode array recording of isolated retinas that are dark adapted to look for retinal ganglion cell function and see if there's a rescue effect there. So in figures A through C, what you see is the response amplitude in relation in response to different stimulation intensity, with the lowest intensity at the bottom and the highest intensity up top. If you look at figures A and figures D, what you see is that with bead injection, without the presence of NLY01 treatment, the response amplitude is decreased. When you do give these animals that have elevated intraocular pressure, NLY01, however, you can see that there's relative preservation of response amplitudes as well as firing rate. Specifically, the transient on response and off responses are most pronounced as being preserved at the higher flash intensities, whereas the sustained on response is sort of preserved throughout the entire flash intensity. It's a little bit of a busy slide, sorry about that. Okay, so given the fact that we know topical administration of GLP-1 receptor agonist was neuroprotective, we want to look for sort of local mediators of its effect. And as I already mentioned, that this class of medication only works through the receptor. Because of that, we needed to look for the receptor in the retina. And the existing literature is really mixed when it comes to this. So we looked at this using two different ways. Number one, we injected a bunch of animals with microbeads, and after injection, we isolated a CD11B positive cell population, which is enriched for monocytes, and we looked for GLP-1 receptor expression and protein in this isolated cellular population, and we were able to find them. Number two, we did a combination immunohistochemistry and C2 hybridization in retinas that were injected with microbeads and therefore had elevated intraocular pressure. And we stain for the RGC marker RBPMS and also IBA1 positive monocytes. And what's interesting is that we found the receptor expression in the IBA1 positive monocytes, but not RBPMS. So this tells us that yes, the receptor is expressed by retinal myeloid cells, but this expression is really only noticeable after intraocular pressure elevation, which is not something we initially expected. So far, all the data I presented have been in mice. So what about humans? Well, we took a subset of postmortem human globes and optic nerve, and we looked for these same astrocyte reactivity markers, the C3 positive reactive, the C3 negative reactive, and the pan-reactive. And we saw that there is a trend towards elevated expression of the C3 positive reactive and pan-reactive markers in the glaucoma optic nerve and retina, suggesting that these same processes that we observed in our mouse glaucoma models also occurs in human glaucoma. So we know that GLP-1 receptor agonists have been used for over 20 years to treat diabetes. So how can we utilize this data to give us an idea as to whether this class of medication may be a good medication for patients who have diabetes and maybe at high risk for glaucoma? So in collaboration with Brian Vanderbeek, who trained with Josh actually at Michigan, we used the Clinformatics DataMart database, which is the same database that Josh had just talked about, which contains de-identified medical claims and demographics information from a large US insurer to sort of answer this question. We selected for patients who are younger than 18 years of age, who initiated GLP-1 receptor agonists, who have greater than 2 years in the database, has had at least one visit to an eye care provider, does not have a prior diagnosis of glaucoma suspect, or ocular hypertension. And in total, 1,961 patients were matched to 4,371 controls. And this was matched based on age, gender, race, and the date of medication initiation. And these controls were initiated on a different class of diabetic medication. What we found was that, yes, GLP-1 receptor agonist use did in fact decrease glaucoma risk for about 44%. If we took glaucoma suspect away from the outcome definition, it actually decreased the risk further with a hazard ratio of 0.08. All covariates were relatively comparable between groups, specifically average hemoglobin A1C and assessment of diabetic complications was comparable between groups. So what can we summarize in this sort of story of GLP-1 receptor agonists that is still relatively ongoing in my mind? Well, we know that this class of medication, which are incretin mimetic that also exert central anti-inflammatory effects, reduces macrophage microglia activation, and this occurs in response to intraocular pressure elevation, and this in turn reduces reactive astrocytes and results in both structural and functional rescue retinal ganglion cells. GLP-1 receptor agonist use is associated with a lower risk of glaucoma in diabetic patients based on our database study. And at the very least, our data suggests that in diabetic patients who are at high risk for developing glaucoma, that perhaps this class of medication should be a preferred second-line agent for treating diabetes if they need something else for their blood sugar control. Okay, so with the remainder of my time, I want to talk a little bit about next steps and what we're thinking regarding sort of pursuing some of the questions that came up in the process of working on this study. So if you recall from a few slides ago, what we saw was that retinal macrophages and microglia do not respond the same way to intraocular pressure elevation. There's a suggestion that these macrophages may respond a little bit faster than microglia. And this brings up the very interesting question of what does retinal infiltration of monocytes, so macrophage extravasation into the retina, what role does that play in glaucoma pathogenesis? So to kind of look at that, we decided to use an MCP1 knockout animal. So MCP1 is a very potent chemokine that is responsible for monocyte recruitment extravasation. In response to injury and disease, MCP1 combined with this receptor CCR2 results in extravasation of macrophages from the systemic circulation into the local tissue that is expressing the injury or the disease. And there is suggestion in the literature that MCP1 levels in the circulation is associated, is correlated with glaucoma severity. Specifically, higher level of MCP1 is associated with worsening visual field progression and also with increased rates of surgical failure. And this is work done by Michelle Guo and Turner Schwartz, which are both medical students at Penn. Okay, so once again, we injected animals, either wild type animals or MCP1 knockout animals with microbeads. And after about 7 weeks, we looked for retinal ganglion cell survival and axon density. And what we saw was that whereas in the wild type animals, both were decreased in the setting of intraocular pressure elevation, they were relatively preserved in the MCP1 knockouts. Further, as you might expect, based on the fact that MCP1 is a potent chemokine, the number of IBA1 positive myeloid cells was also not elevated after intraocular pressure elevation, the MCP1 knockout animals, compared to the wild type. And finally, in addition to structural preservation, MCP1 knockouts also had relatively functional preservation of RGCs. So partially based on our results here, what we want to know is, so here are some of the next steps we're thinking pursuing as part of the R01. So number one, what is the relative contribution of infiltrating macrophages versus resident microglia, right? So as we know, once these macrophages extravasate into the local tissue, their expression profiles almost immediately start to change. So it's very difficult to figure out what is resident microglia, what is an infiltrating macrophage based on expression profile. So the really definitive way to sort of looking at that is through lineage tracing. And so we produce this inducible reporter mouse for macrophage and microglia that expresses a green fluorescence for macrophage and a red fluorescence for microglia. We're going to use that to figure out, okay, what percentage of the activated monocytes are macrophages, what percentage is microglia after intraocular pressure elevation. Secondly, so far I've just focused on the effect of GLP-1 receptor agonists on neuroinflammation. Well, how much does NLY-01 and other GLP-1 receptor agonists exert its protective effects through the receptor on macrophages and microglia versus some other effects, such as the incretin effect that we know it exerts because it's used to treat type 2 diabetes? Well, we made 2 different kinds of mouse, the macrophage microglia specific GLP-1 receptor knockout, and a mouse line that only expresses GLP-1 receptor on macrophage microglia. And with both of these animal lines, we're able to see is receptor expression on macrophage and microglia necessary and sufficient for the neuroprotective effect we observed. And the flip side of that, and finally, this is sort of the topic of this session here, what role does the incretin action of GLP-1 receptor agonists play in its neuroprotective effects? So in order to figure that out, we need to sort of distinguish its insulin releasing effects from its central neuroprotective effects. And one way we're thinking of doing that is by using beta cell specific GLP-1 receptor knockout, which means that these mice are not able to release insulin in response to a glucose bolus because of the release of GLP-1 or GLP-1 receptor agonist use. Finally, we know that this receptor is widely expressed both in the periphery and the central nervous system. So we want to look at what is the central versus peripheral effect of GLP-1 receptor activation. So we're going to do this by using a peripherally acting excitin 4. So we're going to conjugate the excitin 4 to a B12, which will sort of limit its action in the periphery, does not allow it to go into the central nervous system. And this will have direct implications when it comes to figuring out how exactly we can administer this class of medication for treating glaucoma. So is topical administration a viable way of using this class of medications? And that really speaks to, do we need to have a systemic effect in order for this class of medication to work? Or can we just limit its function in the eye and therefore it will be neuroprotective? Okay, well, thank you for your attention. This work would not have been possible without my list of collaborators here, all the people in my lab. Please check out our lab website at qicuilab.org. And for those of you who are Twitter inclined, you can find me at SwayLab on Twitter. Thank you so much.
So I want to really thank Tui for this presentation, because it's really more in the spirit of what we think of for a think tank. This shouldn't be a meeting where we just talk about what's already published and out there, but it's great to see that she's shown what's out there in terms of the literature, what she's doing, and then leaving us with some points to really think about and hopefully that we can discuss later on today. I'm going to invite Colm O'Brien to make some comments. As Josh had alluded, Colm O'Brien's been working on metformin also. He is at the Mater, and I'm going to probably mispronounce this, Misericordiae University Hospital. Did I say that right? Yeah. You did. Oh, because my Irish is really poor. He's obviously Irish.
Other things, Richard.
And so he's from Dublin. Thank you.
Thanks very much, Lou and the Glaucoma Foundation for giving me this opportunity to present and to follow up a bit on the scene that Joshua Stein presented earlier this morning, talking about the potential role of metformin in glaucoma. Metformin can act in a whole multitude of different pathways and different mechanisms. And here we talk about its potential role in lamina cribrosa fibrosis and ECM production in the lamina, but also in terms of its role in mitochondrial dysfunction in glaucoma. So my research group has looked at the mechanisms and pathways that drive lamina cribrosa fibrosis in glaucoma. And we've concentrated on lamina cribrosa myofibroblasts. We're using human donors. And 3 years ago, we published a paper in IOVS using a custom designed platform to look at mitochondrial function. We showed that in that paper that there was reduced oxidative phosphorylation, reduced ATP production, increase in glycolysis, which for those of you who know your cancer biology, this is what's called the Warburg effect. It's been described in cancer cells for a long time, but has also been described more recently in fibrotic cells in different diseases. So we also found increased evidence of other sources of energy within the cell, including glutaminolysis and 1-carbon metabolism. So all of these are part of the Krebs cycle, which, as you've heard this morning, is so important at generating energy within the cells. So sorry, I'll go back. So in this study, we're looking, and our interest in glaucoma was partly driven by Joshua's review paper showing reduced prevalence of glaucoma. But there's other evidence in terms of metformin being antifibrotic in other fibrotic diseases, cardiac, pulmonary, and renal fibrosis. And more recently, in a rat model, a group have published using metformin eye drops that reduces post-operative subconjunctival fibrosis following glaucoma filtration surgery. So that was kind of another driver of this project. So in this study, we used the Seahorse extracellular flux analyzer, which many of you will be familiar with. What we found, and we initially did a dose response curve and found results almost in the opposite way to your study in terms of epidemiological studies, was that the lower dose of metformin that we used was less likely to cause cell toxicity. So we use 0.1 millimolar of metformin. We got this study done in Jane Farrer's lab in Trinity College, Dublin. And this showed us that there was reduced ATP production, reduced oxygen consumption rate, and reduce oxygen spare capacity within these cells. And when we treated these cells with 0.1% metformin, it improved mitochondrial function, increased ATP production, and increased respiratory capacity within the cells. So then, how did that relate to ECM production? We used a number of fibrotic markers, including alpha SMA, which is the key kind of denominator of activated fibroblasts, collagen 1A1 and vitronectin, a number of other ECM genes. And we found that so this is comparing glaucoma and normal, the normal in black, and the glaucoma in the tall bar. So pre-treatment with metformin significantly reduced the expression of alpha SMA, collagen 1A1 and vitronectin in these cell lines. So that gives us the kind of, so we've also looked at downstream other mechanisms within these pathways. I won't go into them because Lou restricted me to three slides, but I've actually gone to 4 slides. So that's kind of a summary of it. But we're looking at microRNAs, in particular micro 26A, which again, with metformin treatment increases microRNA 26A expression, which contributes to the ECM reduction within the cell line. So that's all I have to say, but thanks for the opportunity to present to the group.
Thank you, Colm. So with that, I'm going to open this up for discussion. There's microphones, and you raise your hand. Yeah, you have a better microphone.
Oh, sorry, is that better?
Yeah. I can hand Dr. Baxter the next.
Okay, so I guess my question is, what's next? Is there enough evidence, say, that metformin may be beneficial, that we should be thinking about prospective trials in humans? We, as a community, I think a lot of us felt there was enough evidence for nicotinamide, and there are 4 ongoing trials at least that I know of. Or are there other steps? What do we do next? And given the interest for aging in general, are there any randomized controlled trials for metformin that are happening for other indices of aging that are already happening that maybe we'd be able to sort of piggyback looking at glaucoma on?
Josh, question about the clinical trials?
Is this on? Okay. I'd love to see a clinical trial and on this topic. Clinical trials tend to be very expensive. They tend to, require they tend to take a long time. There may be ways to study this. I don't know. I can think of ways to study this with existing data sets that might inform a clinical trial, and teasing out how metformin is affecting intraocular pressure versus structure, function. That might be useful before embarking on a clinical trial. Obviously, if there was an existing trial that this could be piggybacked onto, that would certainly make it less cost prohibitive. But I think in general, there have been very few large-scale clinical trials. I spent many years trying to get a trial on statins. Lou and others were involved in that. And, despite multiple attempts with the NEI, we weren't able, and to me, pretty convincing evidence, we weren't able to get that funded. So I would love to see a trial. I'm not sure whether the evidence that we have to date would be enough to convince a study section, but I think I'd love to see some additional analyses using existing data sets and potentially leverage that to go for a trial.
Anthony, just to let you know that there's an ongoing trial going on in Hong Kong at the moment looking at metformin. It's a structural study. It's going to look at neurofibrillary changes over a 2-year period. It may not be long enough, and I'm not sure they've actually recruited yet, but there's a plan and a study in place.
I have a question to Josh.
Ann Baxter's next.
Thanks so much for the talks. They're really interesting and informative. One thing I wanted to ask was about maybe a little discrepancy in the results between your two studies that both use the Informatics Data Mart. In Josh's study, it looked like metformin had an association with decreased risk of open-angle glaucoma, but not the other anti-hyperglycemic medications. And then in Chi's study, there was an association between the GLP-1 receptor agonists and glaucoma risk. So I was wondering if you think that is in part due to like the inclusion criteria. You know, in the metformin study, it was age over 40. In the GLP-1R study, it was age less than 18. Is it that people who are on GLP-1R agonist medication have more severe diabetes because they need multiple, medications? I just want to get your thoughts on why the two studies had different results around that.
Yeah, I think you bring up a good point, Sally. When patients are started on these medications, it's not random as to which medication a patient is started on. So presumably, they're first line agent, second line, third line. So you have to be careful that something that's like a third line agent may be, you may get confounding that they have more advanced or more recalcitrant disease. So one of the concerns with metformin is if it is a first line agent, maybe these patients are healthier in some way than other patients, and we're just not able to adjust for that with hemoglobin A1C. And that's in part why I'm a little hesitant to say we should go do a big prospective trial right now. I think doing these big database studies, there's so many potential confounding factors. I mean, a lot of these patients don't just have diabetes, they tend to have metabolic syndrome. So you have hypertension, you have hyperlipidemia. Some patients are on medications for hypertension or hyperlipidemia. All of these factors have been in some way associated with glaucoma one way or another. So to be able to tease that apart, it can be very challenging.
So Alon Harris and then Don Zach.
I think.
That's exactly what I was going to say. You're comparing sort of a first line agent to sort of a second line agent, and the matching process and the way we sort of conduct the studies are also not exactly the same.
So Alon Harris.
Yeah, no, Josh kind of addressed it. I mean, I was concerned with the retrospective analysis in that probably I would guess in these diabetic patients, more than 50% of them are on antihypertensive medications. And, this keeps on coming up in our discussions as major factors that will come up tomorrow as well. And just, I was going to ask how that was accounted for, but you kind of addressed it in a way, so.
Yeah, we can adjust for it in our analyses to some extent. This is based on billing codes and use of antihypertensive medications, but these claims data sets don't have blood pressures, they don't have intraocular pressures. You really need more granular detail, and I think some of the newer data sets that are becoming available may give us an opportunity to tease those apart. It's just very difficult.
Thanks.
Don Zach.
Very interesting session, thanks.
His microphone's not on, I think.
Hello, is it on? Okay. Well, no one needs to really hear it. Now it's on. Anyway, in terms of discussion about metformin clinical trials, just want to mention, I'm not sure the status, but clinicaltrials.gov, there's also a clinical trial for metformin for a form of retinitis pigmentosa going on too. But back to the mechanism, as was mentioned, it's metformin, among other things, is an agonist for AMP kinase. Aspirin is also an agonist for AMP kinase. So in any of the databases out there, has anybody looked at whether aspirin has any effect in glaucoma? Has that been looked at?
I'm not aware of that. A data set like Clinformatics, which was used in both of our studies, captures prescription medications. It wouldn't, over-the-counter medications like aspirin probably wouldn't be captured too well. Using electronic health record data sources, we do keep track of patients who are on aspirin, so it could be possible to explore it that way.
Is that worth doing, do you think? Obviously,
Maybe.
most people take aspirin short term, but there are patients who take it long term.
So it's Andrew Fiola, is it?
Yes, yeah, hi, thank you. Excellent session and very interesting, kind of going back to the clinical question of, is there enough? But I was also curious, not only in this context with obviously diabetes is on the rise, and especially in the US, but if you think about the All of Us and the UK Biobank, I was actually curious about the intake, the type of sugar intake, and if it relates to either the development of glaucoma and those types of things, like where does the sugar come from? Because there's been studies that show there's differences between glucose and high fructose corn syrup, fructose and things like that. Does anyone have any ideas?
I don't think that's an open area that should be looked at, the type of glucose.
It's up there with instant coffee. Utah, and then we'll have Sally after that. Oh, and Sally, then Stuart.
Yeah, so really a fantastic session. So metformin has been used also against aging, and also many diseases study chronic, aging and biological aging. I think Lou also previously mentioned, to do telomere length to measure the biological aging. So I wonder, if the GLR1 receptor, the agonist, and also the metformin can actually make the people a little bit younger against the aging part. So do you think that could play a role there? So I look at the biological markers, all the UK Biobank and the neighborhood, all these have demethylation data. You can predict the biological age part to see if there's anything to play a role there.
Sally Atherton.
I think it's on. Dr. Kui, very interesting talk. Are the animals that you're proposing to make, have you already made them? Do they have other, obviously with the knockouts of the cytokines and things, do they have other systemic effects?
So we have made them. We have not gotten a chance to evaluate everything. The reporter mice should not have any systemic issues that we know of, because these are sort of existing phenotypes that we just sort of put together. It's a Cree-lock system. So I guess the question would be, is that causing some sort of toxicity because of that? So we don't have that answer yet. We're characterizing them.
All very interesting talks.
Thank you.
Stuart McGregor.
Thanks for a really nice session. Lou, I had a question about your talk. So you mentioned about the issue in terms of, you mentioned about the issues in terms of ascertainment bias, in terms of the association between diabetes and glaucoma. You mentioned the genetic studies, which I'm not sure if I was interpreting what you said correctly. I guess there's an issue in that the genetic studies are still based on ascertainment biased glaucoma, and so those MR studies, it's a little bit tricky to know whether that really tells you, whether that removes the ascertainment issue.
Yeah, I agree that's an issue, and that's why I also presented the data from Anthony looking at hemoglobin A1C, where that's not, there is an ascertainment there, and true. That's where the patients with diabetic retinopathy and glaucoma were removed, and there was still a relationship between hemoglobin A1C and retinal ganglion cell thickness. That was inverse.
Yeah, I guess one other thing I would add, is that, I agree that's difficult. We did try, and so in the most recent work that we've done, as you know, we looked at the reverse about whether there was a reverse relationship, and there wasn't, which is reassuring from the point of view of the ascertainment bias, but it's still tricky to know.
Dan Stamer.
Yeah, hi. So it turns out I was in Tucson for 25 years, so I had Gila monsters in my backyard quite often, yeah.
I actually never saw one the whole time I was there.
Really?
Yeah, they're pretty rare.
Yeah, so the data that was interesting is you saw the GLP-1 receptor increase in your glaucoma model. Mm-hmm. Is it possible, but that's because you have infiltrating monocytes that are coming into the retina?
Yeah, I mean, that's our theory, right? So as far as we know from existing literature, the receptor itself is not upregulated. If anything, it's downregulated. So there's no evidence to suggest that, when there is sort of an inflammatory process or something going on in the tissue itself, there is an upregulation of the receptor. That has never been shown. So if that's the case, then what that suggests is that these receptor expressing monocytes are ones from the circulation as opposed to resident ones. But that's where sort of the reporter animals that we've generated come in. We're hoping that will sort of answer that question for us more definitively.
Anthony Kawaya?
So just to follow up on the point about using, Mendelian randomization is obviously a very powerful technique, but it does just rely on the underlying genetic summary results. And as Stuart was alluding to, and this is something we saw, if you take the biggest genetic results, which include UK Biobank, where not everybody's been examined for glaucoma, we're relying on self-report, then you can get those ascertainment biases. And as some of that self-report, for example, just ocular hypertension, I think the clearest example of that is if you do a Mendelian randomization for central corneal thickness and glaucoma, when you've got all of that, a thicker CCT is causal for glaucoma, whereas, all of the studies suggest that maybe a thinner CCT, that could be a collider bias. But putting that aside is doing it the other way around, and that's probably because a thicker CCT will lead to a higher raised IOP, and then you being, sort of diagnosed with glaucoma, and that's driving the association. I wanted to give a bit of context about the study looking at the retinal layer thicknesses and HbA1c. The reason we did that study was kind of not glaucoma driven. It was more that there's growing evidence in people who are interested in how diabetes affects the eye, that one of the earliest signs of diabetes affecting the eye, sooner than microaneurysms, is neuronal, and that's evident in the middle retina and inner retina. So it was kind of looking at it in that way. And there seems to be multiple studies now that point towards metabolic dysfunction affecting the inner and middle retina. Now, if it affects the inner retina, is that glaucoma, or can we affect the inner retina in a way that isn't glaucoma, or is anything that thins the inner retina a tendency towards glaucoma? I don't know. So, I think it's interesting that diabetes affects there, but whether that's necessarily related to glaucoma risk, I'm personally still uncertain.
As sort of a follow-up comment to that, can I just ask, what is glaucoma? Right? Can we actually answer, right? It's a little bit hard to define, and that's the reason I think why a lot of the patients come to us and say, what is glaucoma? What is this thing that we have? And so we kind of give them a little bit of a hand-wavy answer as to what it is. So I think part of that has to do with the fact that we do not have a great definition of what glaucoma is. And the reason for that is because there are all these different pathogenic mechanisms, these multi-genetic risk factors that's impacting glaucoma. So until we kind of know that, it's very hard to define the disease. So it's like a chicken egg thing almost.
Yeah, I agree. And we mentioned before how one of the roadblocks to using AI in glaucoma has been the DICOM
Absolutely.
Compliance issue. But the second is the definition of glaucoma. And we're doing a recall by glaucoma PRS study right now. And so we just call in people, who haven't had an eye, at least they haven't had a visual field ever. And when the majority of them can't do a visual field. And then you start to say, oh, well, we'll just use the OCT. And OCT is way better, but there's problems with OCT as well, with high myopes, and there's all kinds of artifacts. And so you start getting into the weeds as to how can you sort of operationalize a definition of glaucoma. It is a huge problem, and one that we're not going to solve here. And people are trying to solve that, and they haven't succeeded yet. Bob, this is what we have to work on. We have to get the definition nailed down.
So the American Glaucoma Society had a meeting just a few months ago, and the board of directors got together for the new 5-year plan. And certainly this issue of DICOM compatibility and these types of issues for characterizing the nerve is something that was brought up as one of the important issues going forward for the AGS. So there are people really paying attention to that issue. With regard to the comment about, diabetes, I would just leave that and call that diabetic retinopathy, leave it alone. That's my personal opinion, instead of adding to the quandary of what is glaucoma. That's just making messier. And then two questions. One to Lou. When we were talking about repurposing medications for glaucoma, there was a discussion way back about statins. And I'm hearing this thing about metformin. You're thinking, well, is metformin the new statin? And, we left the statins behind. So what was your thinking?
Well, the statin story is pretty complicated. And, when we looked in the Nurses' Health Study, we actually retracted our original finding that there was an inverse association because we made a mistake in the data analysis. And when we controlled more finely for age, the effect went away. And we told the journal about that, and they published the retraction. So then we went back and looked into UK Biobank, and again, we saw no relationship between statins and glaucoma as defined in the UK Biobank. And we looked at the other biomarkers, the interretinal biomarkers, and we didn't see anything there. So this story of the lipids and statins and glaucoma is very complex, and I don't completely understand it. And we need to have a whole think tank meeting on that.
Well, I think the problem is that these patients with the diabetes have hypertension, have lipid problems. It's the whole, how do you separate that? When you're asking, is this a complex disease? Well, it's a complex set of diseases.
Right. And biostatistically, you can control till the cows come home, but there's no real controlling until you actually do a randomized clinical trial. And that's the only way to really get at it.
So then the next question is actually, so you showed that type 1, well, so I could break it down to type 2, type 1, right? Mainly what we're talking here is type 2 diabetes relative to the metformin. But the data that Josh, you showed, the patients who had diabetes who were type 1 were actually protected, right?
They had a reduced hazard of glaucoma.
Okay. So I would assume, since they're type 1s, these are patients getting insulin. And I actually have a handful of patients right now that we've been paying more attention to who are getting insulin drops for neurotrophic ulcers and neurotrophic keratitis. It's been kind of tricky because it probably takes, like corneal neurotization, probably takes at least 6 months, and these patients don't want to wait 6 months. So now they're on, the new nerve growth factor also, so it's confounding what's working. But clearly there's been some good evidence that it increases corneal nerves. And so what do you think that's role is in terms of glaucoma? So I guess Bob wants to answer that question.
Well, no, not that question, but I want to address the comment about diabetes as a risk factor, because I don't know if it's a risk factor.
You might want to bring the microphone a little closer.
Yeah, these microphones, you got to keep it close.
I'll speak loud. Can everybody hear me?
Yeah, but I think that, it will affect the webcast. There's a webcast.
Yeah, there's a
going on.
webcast. So,
Anthony, were you holding this close without your mask? So, a couple of years ago, we published a paper. We were
Can't have the other one.
diabetes in respect to glaucoma. And there was some
Try this other one.
there was some question in the literature. Is it a risk factor or is it not a risk factor? And all the studies are looking at visual fields. So we did something a little different. Instead of using visual fields, we used OCT. We had serial OCT measurements for 1.5 years. And when we looked at OCT, it was very interesting. We found that, in fact, diabetes was not a risk factor for glaucoma. In fact, it might even be protective. And we went back to dissect, who was it protected? And the absolute amazing thing was the patients who were on metformin, it was protecting them from glaucoma. again, using OCT as a metric for looking at progression and looking at risk factors. I think it's fascinating. It doesn't obviate, the need or the use for clinical trials with some of the great work you're doing in Metformin, but don't assume that it is a risk factor. The literature is replete with all these mitigating factors. Ron mentions antihypertensives and all the vascular diseases they have, everything else that's going on. Be very, very careful. We don't know what it's or not.
So I can't remember the sources, but I vaguely remember that diabetics have thicker neurofibrillars to begin with after diagnosis. So that may be confounding, though. It may look protective, but it may be pseudo-protective because for some reason there's some minor amount of swelling or whatever. Am I missing that?
Well, I think what Bob is pointing out is that you have to disentangle the disease from its treatment. And so it is possible that a diabetic who is being treated with metformin is going to look really good in an OCT study because you're actually now treating the root cause of the disease. And I would also say this, the thing that this session has pointed out to me is that the role of neuroinflammation amongst diabetics or patients with insulin resistance suggests that we need new retinal biomarkers for neuroinflammation. We see the Mueller cells are immune cells. And every once in a while, I'll look at an OCT and I'll see neurofibrillary esquisses, but that's not very common. It would be nice if we could have other biomarkers. And so where I'm going with that is at New York Eye and Ear Infirmary, Toko Chui has a way to actually, she thinks she can see macrophages moving around on the surface of the retina. And you mentioned that those cells seem to be moving faster than microglia. So these kinds of endpoints are going to be critical if we're going to design randomized clinical trials, because they might reduce the sample size to get stuff done.
Yeah. So Marie Bell from UC Davis did have a study where she, not specifically to glaucoma, but this is a retinal injury degeneration model where after the injury is induced, you see the either microglia or monocytes or whatever you want to call them just sort of zooming around the retina. They're moving incredibly fast. So they immediately move into sort of the site of injury and do what they do. So I think, we think of these cells as sort of being sedentary, it's just kind of sitting there doing its thing in the retina, but they're actually a very active component of a response to injury and disease. So that part of it is sort of where we think about when this idea of macrophage infiltration comes into play, right? So they're not just sitting there doing their thing, they're moving, they're actively going to the place where they're needed. And I think that definitely plays a part, I think, in glaucoma as well.
So I've lost track who's next, if there was any other questions or comments. Over there.
Or just one other question on the GLP-1 receptor agonist. And one thing that we have come across, I work with a primary care-based diabetic retinopathy screening program. Some of the endocrinologists have expressed concern about Ozempic because of some clinical trial data that showed more rapid progression of diabetic retinopathy in patients taking Ozempic. And just wondering, and when you did your analysis, did you include patients with diabetic retinopathy or not? And whether you think this has any implications for future clinical translation?
Yeah, so there's actually a paper that we submitted that's in review now. I think it was rejected from somewhere, we're resubmitting it again. Also with Dr. Vanderbeek at Penn, where we looked at that specific question, is GLP-1 receptor agonist use associated with higher risk of diabetic retinopathy? And what we, don't ask me to go into the specific details because it's not my area of expertise. But what we found essentially is that it's not a risk factor for diabetic retinopathy if you take into account the fact that there's rapid normalization of blood glucose after treatment. And in previous studies where they show that there is sort of a rapid worsening of diabetic retinopathy. It's all related to the fact that after they are put on treatment, their blood glucose becomes really quickly normalized. And that's what results in the diabetic retinopathy from getting worse, becoming proliferative. So if you take that into account, it does the same thing when they're placed on GLP-1 receptor agonists. So it's not the medication itself, we think, but the fact that now their blood glucose is all of a sudden under much better control.
So I want to thank, I have one more question.
This is something Ted Dreiger told me a while ago when we were looking in this area. He said, and he thought other people had thought this as well, that people with advanced glaucoma, so you've already got primary open-angle glaucoma and then you become diabetic, don't get bad retinopathy. And he thought that, and now when I tried to look in the literature, I couldn't really find much to support that. So I guess my question is, amongst all of the really experienced people here, do you think that's true? And if it is that telling us anything about the link between diabetic processes in the eye and glaucoma risk?
I hear Lou whispering to himself, so maybe just speak louder. He's been having a self-conversation here.
Yeah, my clinical impression is that I've never seen diabetic retinopathy with someone with a cup disc ratio of 0.9. But I don't think it informs the conversation because it's a supply and demand issue, right? So if diabetes is causing retinal ischemia and that's what's driving neovascularization and all the other things, well, if you have less ganglion cells around, the retina is less ischemic, and that's why they're not getting diabetic retinopathy.
So it may be selection bias, but I have a large practice that's shared with my retina colleagues back and forth, and they send me a lot of their diabetics. and this is minus the neovascular glaucomas that come at 5 o'clock in the evening on a Friday. Yeah. Well, any other comments? So I want to thank you all for really a great discussion. It made me really think about a number of things, and certainly looking at glaucoma overall and including diabetes and some of these other complex diseases. And there's nothing worse than being the person holding you back from lunch. So we got to have a few extra minutes for lunch.